Welcome to The Explainer. Today, we are cracking the code of life itself. I mean, the actual blueprint that makes everything from a tiny bacterium to a giant blue whale possible. We're talking, of course, about nucleic acids.
All right, let's just jump right in with a really big question. Have you ever stopped to think, what is the actual blueprint for every living thing? From the smallest little plant to the biggest animal out there.
The answer? Well, it lies in these amazing molecules called nucleic acids. And honestly, the best way to think about them is like this. They're both the architectural plans for a building and the construction manager on site, all rolled into one. They have all the info needed to build and run the entire show. Now, figuring this all out wasn't something that happened overnight. Not at all. It was this incredible journey of discovery that took almost 100 years. It kicked off way back in 1869, when a guy named Friedrich Miescher first found this stuff he called nuclein. And it wasn't until 1953 that Watson and Crick finally gave us that iconic image of the double helix.
Okay, so if we're calling nucleic acids a blueprint, what's it written with? What are the actual building blocks? Let's get into it and deconstruct the letters of the genetic code.
Okay, first up, every letter needs to be written on something, right? In this case, it's a 5-carbon sugar. This little molecule is the structural backbone. It's literally the paper for our blueprint. And you'll see there's a slight difference. In DNA, it's called deoxyribose, and in RNA, it's just ribose.
So we've got our paper. Now we need the ink, and that's where the nitrogenous bases come in. These are the actual letters of the code, A, G, C, and T. And here's something cool. They come in 2 different sizes. You got the bigger ones, the purines, and the smaller ones, the pyrimidines. And trust me, that size difference is super important. We'll see why in a bit.
And finally, component number 3. We have our paper and our ink. Now we need to link the pages together. That's the job of the phosphate group. Think of it like a staple or a binder clip that connects one sugar base unit to the next, creating this long chain. So here's how it all clicks together. It's actually a pretty simple recipe. You start with that sugar, our backbone. You attach one of the bases, A, T, C, or G, and that combination is called a nucleoside. Then you add that final piece, the phosphate group, and bam, you've got a complete nucleotide. And that is the fundamental unit, the single building block for both DNA and RNA. Okay, so we've made our letters, the nucleotides. But how do you go from single letters to this incredible twisted masterpiece, the book of life itself, the double helix? Well, one of the first huge clues came from a scientist named Erwin Chargaff. He noticed something really specific. In any sample of DNA, the amount of A was always the same as the amount of T, and the amount of G was always the same as C. This wasn't a coincidence, it was a rule, and it pointed directly to a very specific pairing system.
And here it is. This is the absolute key to everything. A always pairs with T, and G always pairs with C. They're held together by these things called hydrogen bonds. And remember that size difference I mentioned earlier? This is where it pays off. A big one, a purine, always pairs with a small one, a pyrimidine. It's like a perfect puzzle piece, and it's what keeps the ladder of DNA the exact same width all the way down.
But wait, it gets even cooler. The way these 2 strands line up is really interesting. They don't run in the same direction. They're what we call anti-parallel. The best way to picture it is like a highway. You have the northbound lanes and the southbound lanes. They run parallel, but in opposite directions. And that orientation is absolutely critical for how the cell's machinery can actually copy and read the code. So you take that perfect base pairing, that anti-parallel structure, and what do you get? You get that beautiful, famous twist, the right-handed double helix. It's just an incredibly elegant and stable structure, and it's so consistent that its diameter is always right around 20 angstroms.
All right, we've spent a lot of time on DNA, and for good reason. It's the master blueprint. But DNA doesn't work alone. It has a super important partner. So now let's bring the other nucleic acid, RNA, into the spotlight and see how it's different.
This table really lays it all out, a real tale of 2 molecules. Think of DNA as the priceless reference book locked away in the library. It's double-stranded, super stable, and it's all about long-term storage. RNA, on the other hand, is like the sticky note or the memo. It's single-stranded, it uses a slightly different sugar and base, and its job is to be a versatile, short-term messenger carrying out specific tasks.
And here's the thing, RNA isn't just one guy. It's a whole crew of specialists, each with a different job. You've got messenger RNA, who's the courier carrying instructions from the DNA blueprint. Then there's transfer RNA,
who's like the delivery truck, bringing raw materials to the job site. And ribosomal RNA actually helps form the factory where proteins get built. Plus, you've even got these regulatory RNAs that act like foremen, telling other genes when to turn on and off.
Okay, so we have this incredibly stable blueprint in our DNA. That's great, but think about it. When a cell divides, how does it make a perfect, flawless copy of that massive blueprint to give to the new cell? How does that work?
Well, it happens through this amazing, beautifully coordinated process called replication. It's like a molecular dance. First, an enzyme called helicase comes in and unzips the double helix. Then, a little primer marks the spot to get started. And then the star of the show, DNA polymerase, swoops in and starts building the new matching strand, using the old one as a perfect template. And to finish it all off, another enzyme called ligase comes in and seals up any gaps.
And here's the really brilliant part. This whole process is called semi-conservative. What that means is that every new DNA molecule you make is a hybrid. It's made of one of the original strands and one brand new strand. It's such a clever, elegant system, and it's how the cell guarantees that it makes an almost perfect copy every single time.
So why does all this biochemistry matter? Well, understanding this blueprint isn't just for textbooks. It has completely, and I mean completely, world-changing applications. We're talking about everything from the food we eat to revolutionary new medicines.
When you boil it all down, it comes back to three main jobs. 1. Store the information, which is DNA's role. 2. Copy that information perfectly, that's replication. And 3. Use that information to actually build and run the living thing. That's it. Store, copy, and express.
And knowing this code gives us some serious power. I mean, we can now engineer plants to resist viruses, we can breed crops to survive droughts, and with game-changing tools like CRISPR, we can now go in and directly edit the genetic code, which is opening up incredible possibilities for curing diseases that were once untreatable. Think about it. For centuries, all we could do was observe the results of this genetic code. Now, we can read it, we can write it, and we can even rewrite it. We've gone from being simple readers of the blueprint to becoming the architects. And that leaves us with a truly powerful and profound question for our future. What will we choose to build?